Vaccines have saved millions of lives in the past century. For now, they're the best way out of this crisis, but there are exciting new prospects waiting in the wings. The practice of vaccinating dates back thousands of years, through rabbit spines, powdered cowpox, and fearless scientists. Today, viral vectors and mRNA technology have been instrumental in fighting COVID-19. And another technique could give more hope. DNA-based vaccines. The beauty is that scientists can design them on a computer in a matter of hours. But people are worried about their own DNA being altered. Italy is set to start trials of a vaccine based on a DNA fragment next month. Its drug regulator having approved the COVID eVax inoculation. More on the safety issue in a bit. First, a little history lesson.
As early as the 10th century in China, secretions from smallpox pustules were dried and rubbed into superficial scratches in the skin or inhaled. This is considered the first inoculation using attenuated pathogens to cause a reaction by the immune system. In the 1770s, English physician Edward Jenner discovered that people infected with cowpox, which was relatively harmless, did not contract the much more deadly smallpox disease. In 1796, he vaccinated an 8-year-old boy with pus from a milkmaid's cowpox lesions. 6 weeks later, the boy proved to be immune. Hence the name vaccine, from vacca, the Latin word for cow. Originally, vaccines were based on injecting or administering small doses of attenuated live or dead viruses into the body. The immune system then attacked them and created antibodies, and subsequently an immunity. With more complex viruses, such as SARS-CoV-2, a roundabout method is necessary. That's why research teams try to produce only certain fragments of one viral protein, the spike protein, which, when safely introduced to the body, would stimulate an immune response. Genetically manipulated DNA and RNA vaccines are also being tested. Here, the body produces a protein by itself that triggers an immune reaction. Another possibility is the viral vector vaccine. In this case, a harmless virus is disguised as the SARS-CoV-2 virus. The immune system responds and remembers the characteristics. Side effects of vaccinations are usually limited to a bit of swelling or mild symptoms for a few days. Permanent vaccine damage occurs in very few of those vaccinated. And the belief that vaccinations lead to autism has been disproven by many studies.
Friedemann Weber is a professor of virology at the University of Gießen. Just for the record, DNA vaccines get alarm bells ringing for some people. Just how safe are they?
Well, actually, there's been a lot of studies meanwhile, and I would even say surprisingly, they found very little integration. Some studies just found zero of it, although they clearly found the DNA in the organisms. So apparently they seem to be safe.
But apart from the close to zero results, I mean, is there something to be concerned about there?
Well, yeah, you always have to keep an eye on that, that's for sure. And this is why there's all these safety studies after a vaccine has been rolled out, to just observe for longer term effects if you want. And there's nothing in biology, there's nothing you can exclude to 100%. But just to keep that in a perspective, vaccines, like if you use a DNA virus vector, is in fact also a DNA vaccine. It's just enclosed in a virus shell, but from the point on when it has entered the cell, it's basically the same as a DNA vaccine.
And I guess we can't be 100% yet because they're not ready for humans. They're being tested on animals.
Yes, correct. Yeah, there's 2 animal vaccines out at the time.
What sort of animals and what sort of results have been achieved there?
Well, one is for horses about West Nile virus, and the other is for salmon. It's called infectious hematopoietic necrosis virus, which is a rhabdovirus, well, related to rabies, basically. And in both cases, they found them to be safe and efficient. This is why they are licensed, and you can give them as a vet. So apparently there seems... Sorry?
I was just going to say, if they're safe and efficient and are proving quite successful, why are DNA vaccines lagging behind other sorts of vaccines then?
I think exactly because of the concerns you just expressed. And these are real concerns, no doubt about this, and they have to be checked on and on. But safety is always, during all those clinical studies, the first issue, the first thing to be checked, rather than efficiency, which comes later on. And as I said, so far it seems to be okay. But the other ones are, most of them, apart from the messenger RNA vaccines, are like established platforms. All you had to do was to put your favorite SARS coronavirus 2 antigen on it and let it express by your vector, and then off you go. So that's easier because there's already licensed vaccines on these platforms.
Okay, well, let's compare the mRNA vaccines to the DNA vaccines and how they both work. The DNA vaccine introduces genetic code of a piece of a virus, like the spike protein, for example, into a person's cells, just like an mRNA vaccine, but it then has to get into the nucleus where it's transcribed into mRNA, which instructs the cell to produce the spike protein, priming the immune system for the real virus. So the DNA has to get into the cell nucleus to make the mRNA. Why not just take the mRNA vaccine in the first place?
Well, that's perfectly correct. The only thing is simply messenger RNA vaccines are much more expensive, like 10 times as much at least. So it's also a question of economy. And DNA is just very, very simple to make and very cheap to produce in huge amounts.
It is something that the people are genuinely worried about, though, injecting that DNA into the nucleus of their cells. I mean, can it harm cells? Can it alter the genome?
Well, in fact, if you have a vaccine, you have to harm the cells to some extent, because besides the expression of the antigen, you need to have a little injury to be set in order to alarm the immune system. So this is always goes hand in hand, if you want. And in fact, what we do is we apply the DNA as an electric shock. So you get a little shock, yes, with a device that looks like a pistol. It has a certain amount of electrodes. There's still experiments how many there will be in the end. And after this little shock, the DNA has entered the cell, not necessarily the nucleus. That will be the next step and will not happen in all the cells. But when cells are dividing, the nucleus is dissolving, actually, and it's in the open, and it will kind of, when the cells have divided, form again, and then this plasmid DNA is ending up in the nucleus.
You're making it sound better and better with the electric shock and pistol, but tell me, how will a DNA vaccine...
I've seen videos of people, they survived it, yeah.
They survived, that's good to hear. Tell me how a DNA vaccine could actually deal with something like mutations, which is on everyone's minds right now.
I think with a DNA vaccine, one is as flexible as with a messenger RNA vaccine or with a adenovirus vector, for example. All you need is your new sequence. Either you insert, if it's just one mutation, you can insert it like by hand in the lab. Everybody could do that in the lab. Or if it's more mutations all over the gene, then you just exchange the whole gene. But this is basically normal lab work and not much of a problem.
You heard it from Friedemann Weber, a professor of virology at the University of Gießen. Thank you very much.
You're welcome. Thank you, Ben.
Let's get you over to our science correspondent, Derek Williams. He's been looking into your questions on the coronavirus.
How does the Pfizer-Biontech vaccine work?
I've answered this before, but the question is posted again and again, so I guess it's maybe time to go over the details again. Two of the vaccines that have now been widely approved, one developed by Biontech and Pfizer, and one produced by Moderna, are what are called messenger RNA, or mRNA vaccines. Now, the technology behind them has been at the focus of a lot of research for decades, but this is the first time they've received widespread approval from healthcare authorities. Unlike vaccines based on traditional platforms, ones that, for example, use inactivated versions of the virus, the Biontech-Pfizer vaccine leads to an immune response in the body by delivering information in the form of a special molecule, messenger RNA. mRNA molecules are single-stranded chains of what are called nucleotides that fulfill a very important function in cells. They're kind of the blueprints for making proteins and act as messengers between a cell's headquarters in the nucleus and its protein-building factories out in the cytoplasm, hence the name. But the protein these new mRNA vaccines encode for isn't a human one. It's a protein made by the coronavirus. And when that mRNA is injected, it causes your cells to begin making that viral protein. And that viral protein provokes an immune response, just as if you'd caught COVID-19. Sending in the pretty simple, quick-to-produce mRNA code molecules to make these complicated proteins and getting the body to do the work is a great solution to a complex problem. And there are high hopes that mRNA vaccines, which seem to have finally come into their own, are now set to revolutionize a range of fields in medicine.
And keep sending in your questions to our YouTube channel. Thanks for watching, stay safe, and see you again soon.